Okay, good afternoon, everybody, and welcome for our DMG weekly seminar series at the Department of Marine Geosciences. And today we're hosting Dr. Hitisha Baroliya here from our department. She's a postdoctoral researcher at our department, and her research focuses on marine biodiversity, ecology, and biogeography with over 8 years of experience in marine ecology and species distribution studies. She is currently working on a National Geographic funded project titled Diversity and Biogeography of Recent Shallow Water Foraminiferal from the Maldives, cataloging species diversity and exploring foraminiferal assemblages as baseline indicators of reef health to support marine protection and policy planning. Dr. Baroliya earned her PhD in zoology from Saurashtra University in India and has authored several peer-reviewed publications and presented her work at international conferences, including at the EGU in Vienna and MECOS. So, welcome, Tisha, to the seminar. And today she's going to talk about her research in reef Earth through the foraminiferal lens, a baseline for marine protection planning in the southern Maldives. So thank you very much. Appreciate the foreign viewers.
Thank you, Professor Nicholas, for the brief introductions. So, hello everyone. I'm Hitisha Baroliya, and today I'm talking about foraminifera perspective for the reef health. Usually, coral reefs are tells their story through the corals and the fishes, but today I want to show you how what happened when we listen reef through this tiny marine organism foraminifera, which tells about which reveals the pattern of their stability, their stress, and their resilience. This study is the part of the broader project National Geographic Pristine Seas and Pristine Seas program. And to for this for this study, I present into two phases. Like first, we I'm talking about the baseline in this data poor region of southern Maldives, and then I will talking about how this baseline database useful for this marine protection, like how foraminifera can inform the integration into the marine protection planning regions. So, for first, I would like to begin with the briefly introducing the broader project about the pristine seas. Like Pristine Seas is a National Geographic program, which main aim is to protect the last. Main aim is to protect the intact marine ecosystem with the science-based conservations. And within this broader project, our involvement is like our main role is to focus on the sediment collections and do the foraminiferal analysis. And to complement their fish, their benthic invertebrate fish and megafauna surveys. Our contribution is not much so far like broader consequence, but it is like extremely informative for the long term monitoring program. I know many of you aware about what is the foraminifera, but I would like to be free background giving. I would like to give brief background of the foraminifera because it's a main. This tiny marvel is the main hero of this whole talk. And like a foraminifera is a single cell protist built up from the calcium carbonate, and it is highly abundant in the marine sediment and contribute their carbonate production. Here you can see the several photographs of the different types and shapes of the foraminifera, and this foraminifera is like extremely sensitive to any kind of the environmental changes, which make them a valuable and important candidate to detect any ecological shift in the particular environment. So within this context, you can see this in this map there is different locations where pristine seas from the where sediment was collected during the Pristine expeditions. And all of the most of their area is very remote locations where the biological monitoring is limited. And within this like for this the Foraminifera database ecological baseline helpful for this data poor regions. And this the marker here shows that the what kind of the progress or what kind of the results we have from the particular locations. Like the red marker here shows that we have preliminary results of that area regarding the ecological baseline and their surrounding conditions. The yellow color marker shows that the advanced kind of the results, and the white color marked area shows that we have completed our foraminifera analysis and created the ecological baseline for the foraminifera database. And from the Salva genus and new, this result are already published. I am currently working on specifically the Maldivian region. So specifically in the Maldives, we focused on the southern part of the Maldives. The southern part of the Maldives, we because there is no any foraminifera database from this region, and the second is that now the Maldivian government implement expansion of tourism from the to the southern regions. So within this context, we examine the three atolls like Huadu, Fuamula, and Adu atolls. These three across three atolls, we have the 26 distinct localities from where the sediment samples were collected. All these atolls are different in the geomorphologically, their exposure, their human pressures, and also the marine protection area selections. In this map, you can clearly see there's the green line shows the marine protected area, and each color is designated for particular atoll. Like the blue color is for Havadu, red for the Fuamulla, and the orange color is for the Adu atoll. And also there is different shape which represent the what kind of the substrate present over there. Like the round shape represent the algae, square shape represent the shelly substrate, and the triangle represent the coral substrate or the coral habitat. So this will be constant throughout the presentation. As we know about that, like Maldives is mainly famous for its tropical reefs, pristine beaches, and the vibrant marine life. So for that, within to keep this in the mind, the main aim of this study is to create a robust ecological baseline, and how this baseline can help us to assess the reef health, and also under them also tells a cost effective also foraminifera can be a cost effective tools for the biomonitoring purposes and for the long term monitoring. So for this study, samples were collected using scuba diving, and one of our partner from the Hawaii Institute, Dr. Alan Frendler, did the scuba from all the 26 sampling sites and collected 100 ml sediments, which further preserve in the ethanol with the rose bengal. And to do foraminifera analysis, we follow the standard protocol, like to take 10 ml subsamples. Then we sieve these using the 63 and 125 sieves, and then dry the samples and pick approx 250 foraminifera from each of the reef sites, and then identify the species and just cataloging them to just cataloging to describe what they are, like what kind of the biodiversity. And using this database by creating diversity and their assemblage, we assess like what kind of the environmental, what kind of their current conditions, current reef health state. We further use them to integrate into this. We also do statistical analysis using the R software, and we use different statistical for biodiversity. We use the species diversity index, and for the ecological interpretation for the ecosystem perspective, we use that cluster analysis, then NMDS, and all this before prior to any kind of analysis, the data will be normalized. So before begin to talking about foraminifera diversity, I would like to give you the general sedimentary context in the southern Maldives. Like here you can see in the photographs, the sediments are rich in the carbonate, like with the foraminifera, then the other coral fragments, then the algae fragments. And why, and this sedimentary context is important because this shapes what kind of the biological foundation they have in the southern Maldives. So across the 26 sampling site, we identified 320 taxa belonging to 118 genera. And here in the plate, you can show in the left side of the plate, you can see which is the most dominant and the most common species. And in the right side, you can see the species which is less common but still have the greater than 1% abundance in the total assemblages. So this suggests that it's a relatively very high, comparatively higher biodiversity in the southern region of the Maldives. Also, they have these in this plate. I'm showing you some of the rare taxa, which is like under 1% of the total abundance, but it is not. It only contribute little to the abundance, but it is extremely important for the biodiversity perspective, and also very vulnerable to or sensitive or any kind of ecological change. So we are so we want to give not only emphasis like what kind of the presence or absence of generalized catalog in the foraminifera diversity, but we like to interpret into the ecosystem level. So, and we also want to check like how this species like how this baseline can give us the information about the total environment. And for that we did some further analysis like diversity indices, then community compositions, and what kind of the sharing across all these reef habitats. So here in this graph, you can see that the species diversity, richness, and the evenness index across the three of the atolls. And it suggests that the diversity is relatively high, and also all the species are evenly distributed within each of the substrate and the each of the atoll. So which suggests that the foraminifera diversity in the southern Maldives is really indicate very stable and uh, really stable reef health conditions. But when we look at the community composition of each of the atoll, like we found that only one-third species are shared across each of the atolls. So which suggests that which might indicate that this species is each of the atoll is in work like as a semi-unit, not like a general homogeneous condition in a reef level, or like a uniform condition to the reefal system. To further understand this and to test whether this is how their foraminiferal diversity, especially variations, is looks like, we further do the cluster analysis and the NMDS. And in the NMDS, we use the most top 10 dominant species vector, which gives us the information like what species really drives these spatial separations. So in the cluster, we clearly see that there is two distinct clusters. Like one cluster shows that mostly clustered together, it's the Hawadu cluster, and the another cluster there is mixing of the Adu atoll and the Fuamula atoll. And in the when we look at the NMDS, it's also clearly indicate that the Havadu is tightly clustered together, which and also this whole assemblage is driven by the stable, well lit reef symbiotic reef taxa like Amphistagina and Anamolina, so which indicates a very stable or more pristine or healthy kind of the conditions in the Havadu area. Further we look at the another tight clustering by the Fuamula atoll, and it's most of the taxa is like planktonic and the higher energy taxa. Their assemblage is dominant by this kind of the taxa, so which indicates their they have the separate assemblage, and those indicate some kind of the higher energy state and some transitional kind of the state over that. And the last, the Adu represent the widest dispersion in the NMDS, and it shows like it has the all this from the healthy sites to some kind of the ecological stress kind of signals. Because if you see in the cluster, there is site number 18 and 26 are clustered separately together, and when you check here in the NMDS also, the higher quinenculina represents at this specifically these two sites. So this also give us information like it is balanced situations, like they have some kind of the healthy reef, but it the forum with the help of foraminifera we can also say there is something early warning of for their environment. Further, to check this further to check for the each of the atoll level, to check how this diversity or how this assemblage is variable with reflect with each of the other environmental factors like the hydrodynamics, then the depth, then the what kind of the reef types, and the what kind of the habitat types. We first took a look at the atoll level, and the atoll level we find that it's not it's a marginal significant. But when we check the species turnover, it like here in the graph B, you can see that the species turnover is highly significant between the atoll, which again gives us a strong confirmation that the foraminifera assemblage over in the southern Maldives is strongly structuring by the regional impact. And here in the figure C and D, it represents the current and the wave pattern respectively, which is not directly influence the foraminifera assemblage or the diversity, but it shows that the moderate current and the lower wave pattern supports the reflects the higher diversity, which further indicate generally very stable reef environment. When then we took into look into the what kind of the reef state they are occurring and what kind of the substrate, and also deeper into the depth. So we found that the stronger driver of the this assemblage and the diversity is the what kind of the substrate they occurs. And also if we see the depth is negative weakly relate to this diversity. But when we divided depth into the subdivided into the substrate level, we found that only the coral substrates shows that the positive relation within the depth. And these things also further confirm with the like the coral substrate supports the higher diversity of the foraminifera. So within this context, this is extremely important to understand, like the for the foraminifera assemblage, the main driver is particular substrate. So rather than protection, giving protection to the whole area, it is extremely important to look at protection substrate by substrate or for the particular habitat zone. So till now, we have the key findings, like overall the it suggests that we have very the southern Maldives has very good biodiversity, very good biodiversity, and comparatively higher than central and the northern Maldives, and which indicates the but it shows the clear separation from the intra-atoll level heterogeneity. And most of this dominance by the symbiotic benthic foraminifera and the planktic taxa, which indicates stable reef. And also some of the sites gives us a detectable or the early signal of some changes, which now this finding leads us to our second questions, like how we can integrate foraminifera as a tool for the reef health. So for this, by examining the balance between symbiotic taxa with some opportunistic and stress tolerant taxa, we can assess the foraminifera can help us to test about something for the reef health. So we use for this we use only the dominant and the informative genera from for the southern Maldives, and we divided health state like the pristine, healthy, and transitional based on the indicator taxa we have in a southern Maldivian database. So for this, first we look at our database, like for the genera level, like what kind we have in the greater than 1%. Like we have around 118 genera in overall total assemblage, but only 18 genera represent this greater than 1% across all the study site. And in figure B, you can see their spatial variations at each of the sites study site level. And in this dominant by the amphistragina and followed by the quinenculina. So of course amphistragina give us idea about like very healthy and well lit kind of reef conditions, but the quinenculina shows some patchiness and some increase at particularly some sampling sites, which also gives some stress kind of the conditions at the particular sampling station. So based on this, we divided our samples into the four. We divided our site into this four reef health category, like pristine, healthy, transitional, and impacted. So pristine is like dominant by symbiotic taxa and low representation of the stress tolerant like quinenculina, bolivina. In healthy sampling site, we grouped only those site which has the high but balanced symbiotic taxa with moderate representation of the opportunistic. In the transitional, we group all the site which shows that the lower diversity of lower representation of the symbiotic and the higher of some energy taxa and some stress tolerant taxa. And impacted area, like it is defined by the uh, dominance of the opportunistic taxa and with low or absence of the symbiotic taxa. So for this, we again map the cluster analysis and the NMDS, and we see that for this time we consider only those species which contributes like greater than 5% in the total foraminifera compositions, and we found that out of 320 taxa, only 19 taxa is contributed like greater than 5% of the total foraminifera community. So here we group site based on each of the here we grouped or clustered based on the each of the health reef health category we already described earlier. And here this is also I'm showing here in this C, in the graph C, the relative abundance of each of the six selected genera which contributed in greater than 5% and the more than also the greater than 1% at the genera level, like Amphistragina, Bolivina, Calcarina, Glabraterella, Homotrema, and Quinenculina. These are also, this all genera are very much informative for the biomonitoring purposes in the, for particularly for the reef health. Like Amphistragina indicates good water quality and uh, light condition and temperature conditions. In contrast, the Quinenculina, Bolivina gives us signal about what kind of what kind of the stress over there. And then further we spatially connected we spatially see them on the mapping. Further we map on the spatial connectivity to give in to connect this into the for the management purpose. Here this pink color indicates the pristine sites, purple color represents the healthy sites, yellow color represents the transitional, and the red color shows the impacted sites. So if we see how do atoll represent most of the pristine and the healthier reef health sites, while the formula is considered most of are the transitional state. So also there is two sites which comes under the marine protected area. What makes Formula so special is that it's a single island atoll and with most of the human populations, like with it is like with inhabitants, but in the Havadu most of the area are the inhabitants. And in the Adu atoll, it's a mixture of all the sites. The site number, the middle of the sites, like 21, it's only the pristine sites. While if you see the another sites like site number 23 and 25, which comes under the marine protected area, it comes under the healthier category, while the site number 22 and 24, this is also represent from the coral substrate, but it's the comes under the transitional sites. So this from by the foraminifera we can give us the we can give this informations like the foraminifera sensitivity and offer like foraminifera can offer a scalable tool for the marine biomonitoring. And also the site number 18 and 26 is like extremely important. Like if you see the in the graph B, you can see the uh, relative abundance of each of the selective taxa, like from higher Amphistragina to the lower higher Quinenculina ratio. And if you see the impacted area side, there is a highest Quinenculina and the lowest representation of the Amphistragina. So based on this database, all these sites are categorized, and This image is further analog our foraminifera indicators, like what kind of the this photographs is the representative from each of the sites based on the category. Like in the pristine areas, there is very balanced growth of the algae and stuff. Algae, like in the healthy, there is still balance and this distributed of the environmental conditions. But in the transitional, you can see there's the very sparse growth of algae on biogenic structure, but it's also increased some dead shell structure. And also, if in the impacted area there is completely vanish of the algae, so there is so this supportives our foraminifera indicators based reef health site. So overall, this study shows that still in the current time, Maldives, in the southern part of Maldives, exhibits very stable ecosystem, and also like clear separations in their biogeographic and the spatial variations. But some of the area are in the giving us the early signal which we cannot see by our for the reef health conditions, like the coral degradation before it happens. And yes and oh so this gives this brings me the final conclusion that this foraminifera can integrate into the marine protected planning and give the complementing to the fish and the coral based survey. And also the foraminifera offers the scalable sensitive, scalable, and cost effective tools for the marine protection planning. So thank you everyone for the listening, and if you have any question you can ask.
Okay, thank you very much, Tisha, for the enlightening talk. It was really enlightening for us. So also the young students will know what our more experienced postdocs are doing, right?
Yeah.
So I'm opening the podium for questions from the audience. So feel free to ask and just jump in. We don't see all of you
Uh,
on the Zoom. Yeah, please,
hi,
Alexandra. Yeah.
Nicholas. Hi, Hitisha. It's a really amazing presentation. I have a couple questions. First of all, it's about methodology. As I understood, a Pristine Sea project has established one methodology for different projects. I mean Maldives. Yes, is it?
So
I have one question. Why you use CIF63 for your project? Because it's for evidence of large benthic foraminifera is like
yes,
expected. So 63, it's a bit
it's a bit challenging to pick, and I to pick 63, but it is like um, actually, Dr. Shai set this protocol for particularly the Pristine Seas project, who published material from the Salva genus and the NEO. So I just followed her protocol for my study in the Maldives to, because from for this, like our mainly focus within the Pristine Seas, our focus on the sedimentations and the foraminifera analysis. So for now, we all focused for the all of this area, we use this like 63 and 125 mmcu size for to make a statistical robustness for the strong comparative for overall assemblage to compare the different biogeographic areas.
Thank you. Nice. And another sorry, another question about sites 18 and 26.
Yeah.
It's a really interesting result because Bolivina and Quinculiculina seminula, they are like stress tolerant, yes. And
Yeah.
what do you think these two sites they impacted by human action, or maybe it's a natural sites where, for example, I paid attention that 18 site, its location like semi-close,
Yeah.
and maybe this is like their more dominant silty substrate or no, I don't know. So my question is it impacted by human, or maybe it's a natural stress condition?
So we are still looking on it, but when we look on the Google map around what kind of the because Maldives is famous for its resorts island, so this site 18 is like actually a resort island. So maybe there is the human like I don't know, like vessel trafficking or this kind of the things might influence that area, as well as the nutrient enrichment, like the excessive sewage through, because this site 18 and 26 both are near to one of the resorts. So this might be other possibilities. So, but still we are figuring out to linking like how population per site influence this our results. So we are working on it.
Thank you. Thank you.
Thank you, Shasha.
Any more questions from the audience?
If there's no questions, I'll ask one, but all the questions are coming from our lab.
Yes.
Hi, thank you for the talk, Hitisha. I'm sorry I missed the first part of it. So you might have talked about this, but can you say something? There's the Calcarina curiosity with looking at the movement of Calcarina. Is there anything you can say from your samples about that species and the impacts it's had?
Yes, like Calcarina is kind of invasive species, but it's not in the Indo-Pacific regions. It is in Caribbean and other part. The Calcarina is, but I'm also surprising when I'm look at the data from the Central Maldives and my region, Southern Maldives. So in the Central Maldives, the Calcarina is the higher percentage compared to what we have in our database. So I'm still figuring out about it, but I have no any clue. But one thing for sure, like it's not invasive to the Indo-Pacific area, like Indian Ocean region.
It's not endemic, you're saying. It's definitely not part of its normal assemblage.
Yes.
Thank you.
Any more questions? Well, I think that there are no more questions, Tisha. So thank you again, and feel free to approach her if you may have questions or whatever. She's based at Beverly's lab, and you can contact her directly. So I wish you a good continuation of the day, and see you, I think, in 2 weeks. Next week there is a full seminar.
Thank
Okay?
you. Bye.
Okay. Thank you. Bye bye.
Bye.
Thank you.
Thank you.